Hello everyone. So the next topic which we will going to study is about the electron transport chain. So let's start with the definition. That it is a common pathway which is in the aerobic cells by which the electrons they are derived from various substrate and they are transferred to the oxygen. So ETC, it is a series of highly organized oxidation reduction enzymes. So it is a chain that is highly organized with the oxidation reduction enzymes. What is the location of ETC? ETC is being localized in the mitochondria. So mitochondria, they are the centers for the metabolic oxidative reaction to generate the reduced coenzymes, and they in turn are utilized in the ETC to liberate the energy which is in the form of ATP. So they liberate the energy in the form of ATP, and the location here is the mitochondria which is also called as the powerhouse of the cell. That is why here the energy which is released is in the form of ATP. Then if we go with the introduction that what is electron transport chain, then it is a series of redox reactions, and they resemble a delay race of or bucket bridge in that electrons are passed rapidly from one component to the next. So they are passed from one component to the next. So the electrons, they reduce the molecular oxygen and they produce the oxygen. So electrons reduce the molecular oxygen and produce the water. Now this is a diagrammatic representation of the electron transport chain uh, in the mitochondria. So this is a intermembrane space and this is a mitochondrial matrix, and this which is being labeled, it is the inner mitochondrial membrane. So it is a series of electron transporters which is embedded in the inner mitochondrial membrane, and the electrons from NADH and FADH2, they are converted to molecular oxygen. So here in the process, the protons are pumped from the mitochondrial matrix to the intermembrane space and the oxygen is reduced to form the water. So here oxygen is reduced to form the water, that is H2O. It is being formed. This is cytochrome C where the process is being taken place. So this was all about the electron transport chain, that how the mechanism of electron transport chain is being divided. Now the electron transport chain has few complexes also. So we will talk about these complexes. First is complex 1. So complex, to start with the complex 1, there are two electrons which is from the diagrammatic representation itself, it has been shown that how the process of electron transport chain works in complex 1. So to start with, two electrons, they are carried to the first complex aboard NADH, and this is labeled as complex. So labeled 1. So complex label 1 is being there, which is composed of FMN and an iron sulfur containing protein. So FMN, which is being derived from the vitamin B2, and also fiboflexin, which is one of the several prosthetic groups or cofactors in the electron transport chain. So it is a non-protein molecule which is required for the activity of a protein. And complex 1 can pump 4 hydrogen ions across the membrane from the matrix into the intermembrane space, and it is in this way that the hydrogen ion gradients is established, and they are maintained between the two compartments which is separated by inner mitochondrial membrane. So this was about the complex 1. Then about the complex 2. Q and complex 2. Here complex 2 directly receives FADH2 and it does not pass through any complex 1. So the FADH2 in this will not pass through the complex 1. So there is a compounding connecting, there is a ubiquine which is a compound which is connecting the first and second complexes to the third. So this is the Q which has been labeled as. So Q molecule is a lipid soluble and freely moves through the hydrophobic core of the membrane. So once it is reduced, QH2 ubiquitin delivers its electron to the next complex in the electron transport chain. So ubiquitin, it has been delivered its electron to the next complex in the electron transport chain. Now complex 3, which is composed of cytochrome B, another FES protein, risky center, and cytochrome C complexes, and this complex is called the cytochrome oxyreductase. As in the figure, there was a depiction of STC, that was cytochrome C. So here the role of cytochrome C comes in the picture. So cytochrome proteins, they are a prosthetic group of heme, and this heme molecule is similar to the heme in the hemoglobin. So it carries the electrons but not the oxygen. So the oxygen is not being carried out. It only carries the electrons. So as a result, the iron ions at its core, it is reduced and oxidized, and it passes the electrons, flaunting between different oxidation states, that is Fe++, that is a reduced state, and Fe3+, that is an oxidized state. So this was about the complex 3. Now complex 4, it is composed of cytochrome proteins C, A and A3. So these are all the cytochrome proteins. Now this complex contains two heme groups, one in which of the two cytochromes A and A3 and the three copper ions, a pair of CuA and one CuB in the cytochrome A3. So these cytochromes basically hold an oxygen molecule very tightly between the iron and copper ions until the oxygen is completely reduced. So this is what happens in case of the complex 4. Now the next process is chemoosmosis, in which the free energy from the series of redox reactions, they are just used to pump the hydrogen ions across the membrane. This was all about the electron transport chain. Now coming to the conclusion part, electron transport chain is a portion of aerobic respiration. It was already been described in the definition part that it is a portion of aerobic respiration, and it uses free oxygen as the final electron acceptors of the electrons, which is removed from the intermediate compounds in the glucose catabolism. So the electron transport chain is composed of 4 large multi-protein complexes, which is embedded in the inner mitochondrial membrane, and 2 small diffusible electron carriers shuffling electrons between them. So the electrons, they are passed through a series of redox reactions with a small amount of free energy used at 3 points, which transport the hydrogen ions across the membrane. So this process, what it does is basically it contributes to the gradient used in the chemoosmosis. That was the last step we studied. So the electrons that are passing through the electron transport chain, they gradually lose the energy, and there is a high energy electron which has been donated to the chain by the either NADH and FADH2. It was being represented in the figure in a diagrammatic representation that how this process was going on to complete the chain, and as a low electron energy they reduce the oxygen molecules to and form the water. The level of free energy of electrons, they drops from about 60 kilocalorie per mole in NADH or 45 calorie per mole in FADH2 to about 0 calorie per mole in the water. So the end product of the electron transport chain are water and ATP. So these are some of the end products, that is energy and the water. So a number of intermediate compounds of the citric cycle can be diverted in the anabolism or other biochemical molecules such as non-essential amino acids, sulfur and lipids. So these same molecules can serve as an energy source for the glucose pathway. So this was all about the electron transport chain. The end products of electron transport chain are ATP and water. Then we have described the NADH and FADH2 in the figure diagrammatic representation. So this was all about the electron transport chain. This is the reference.